But exchanging DNA is not reproduction necessarily. It's just, however, when the cell that has exchanged DNA reproduces, then you hope that it's going to reproduce that new DNA that it got. That's what we're hoping our E. coli is going to do now. It's going to reproduce and be resistant, So that's kind of, we're trying to understand it ourselves by doing the actual experiment, because then you can, I think, understand it much better.
Yeah.
And we may fail completely. We may have done it completely wrong. It's the first time we've ever done it, Did it without, any kind of scientific instruction, because I think it's really, it's sort of like do it yourself, kind of learn it yourself. And of course, this is an experiment that's been done a million times. It was done early in the 70s. It's the classic recombinant DNA experiment. So we're learning it, because it'll be a basis for doing other things.
This is what's called streaking a plate with bacteria.
Isn't that dangerous to touch it?
No. I'm not touching the bacteria, I'm touching the plate.
Right.
But this is actually a standard lab experiment that a lot of high schools do in their biology labs. And it really follows all of the principles of cell culture. This is a sealed, store-bought carton of organic yogurt, right? This is going to be my starter. And it says on it that there's 6 live cultures in here.
So 5,000 years ago minimum is when we were making yogurt as a human civilization. Part of the same process that she was doing over there is going to be done over here. It's what we call the scientific method.
And the lignant ones, they also excrete a kind of digestion enzyme, and that allows it to sort of eat its way. You wanted to be something inner cells start to cleave and split apart. And those cleavages can be separated and they can become whole new organisms. They can also be, because they're pluripotent, they have great value because they can also be teased and turn into basically any cell in the body, whether it's lungs, liver. So those cells are very valuable, especially in the early stages of cleavage.
We splice it in. That's the
recombinant.
The topic of this experiment is gene splicing. We're taking 2 proteins and adding them to the genetic material of the E. coli, and now magically the new bacteria that we will grow on the petri dishes, will be resistant to the antibiotic.
Streaking is a process by which we spread cells over the surface of a nutrient medium in order to let them grow.
This is a plate that we streaked with E. coli bacteria. You can see a lot of growth here, right? You can see this white stuff. That's what's basically in your guts, right? And it was streaked on a plate that had no antibiotics in it.
Sort of rub this back and forth like very lightly. Like that. You just touch the surface. It's very much like jelly, and you don't want to break it. See there, I kind of accidentally dug into it. You don't want to dig in, you just want to evenly distribute it on top of the jelly. Go back and forth that way.
Really feel like you're spreading like a very soft little,
Vectors is a DNA molecule that can be used to carry foreign DNA fragments. In other words, these are things that we can change. We can change the DNA of these little vectors and put them into the cell. Yeah, there's a lot of them. And, they're little bits of DNA that are floating around in the E. coli. See, bacteria is really an interesting organism because bacteria exchanges DNA with other bacteria all the time. So scientists have figured that out, and so they're just kind of in a way copying, in the lab and directing in the lab what bacteria naturally does.
E. coli and its metabolic processes in our gut helps our body make that vitamin so that we can have healthy blood clotting go on. If this person has a cold or something and is taking antibiotics, the antibiotics go through the whole system and they're going to kill the normal flora. And so that's why we want resistant bacteria. That's why we could live with resistant bacteria. We live without it, and yet if we do take that antibiotic and it kills a lot of our E. coli, what happens then is something we all know and don't love, diarrhea.
Plasmids, which are basically little process. I want to learn about the process because it's like, yeah, because it's a really basic process of biotechnology. We're trying to learn this experiment so that we can understand it better. But basically, the plasmid has actually areas on it that are called cloning sites that can be broken open.
As a scientific group is, it was actually done it for the first time in '74 by 3 scientists in the lab working with E. coli, and they split parts of protein